So welcome everyone to the guest lecture of Machine Learning 1 course. Today we have with us Dr. Rajat Mani Thomas, and it's a great privilege to have him. He's going to speak on artificial intelligence for medical imaging. For those of you who have some affinity towards using AI for medical imaging, I guess this is going to be a very exciting presentation from him. So first of all, I would like to thank him for taking out time for this presentation. But before we go to his presentation, I would like to introduce him. So we have the speaker here. He's an assistant professor at the Amsterdam University Medical Center in Netherlands, and also he's a AI lead at Circuit Technologies Amsterdam, Netherlands. And to provide a brief biography about him,
Biography about him.
Dr. Thomas
Dr. Thomas.
completed his bachelor's degree in electronics and communication engineering with focus on signal processing from Bangalore, India. He worked at the Raman Research Institute and was involved in the development of deconvolution algorithms for radio astronomical images. He later pursued a master's degree in space technology and radio astronomy at Chalmers Institute in Sweden. Then he worked at the Max Planck Institute for Solar Systems, where his research focus was on building genetic algorithms for detecting extrasolar planets. With his interest in this field, he went on to pursue a PhD in computational astrophysics in Netherlands. During his PhD, he studied the early universe evolutions and how black holes affect it. Dr. Thomas later pursued postdoctoral appointments in Tokyo in Japan and Toronto in Canada in the field of astrophysics. Later, he switched to the field of cognitive neuroscience in Amsterdam and has been working on the analysis of fMRI, EEG, and ECOG data. He is currently working on advanced deep learning based medical image analysis. He is also leading the AI team at a startup in Amsterdam, which is focused on using AI for agriculture. So as you can see, he has a diverse set of experience with different domains of science as well as deep learning. And I guess we'll get to know more about him as well as his experience from his lecture. So welcome, Dr. Thomas, and we are looking forward to your lecture. Thank you.
Hello. Thank you. Thank you for the introduction. Just a request. There is someone who's not muted. There's a lot of background noise.
Yeah,
Could
so it would be
all
nice
of
if
you
you
mute?
could. I'll.
Okay. Am I still visible? Sorry, visible. Audible?
Yes, you are audible. Maybe you can share your screen. I can stop mine.
Yeah. Okay. Can you all see my screen now?
It's visible to me.
Okay. Great. So first of all, thanks again, Deepak, for inviting me. This sounds really interesting, and it's my pleasure to talk about the work I've been doing in machine learning and artificial intelligence in medical imaging. And I hope I can inspire some of you to start thinking about this field and thinking about if you'd like to work in this field in the future. Yeah, I mean, listening to my own biography, it felt like I was doing more astrophysics. That is kind of true. I did a lot of that until 5, 6 years ago when I really switched to medical imaging. And today I'm going to tell you mostly about some of the work that we have been doing. And there are some works in which I've also collaborated with Deepak in this presentation. Uh, but mostly I want to provide you an overview of how machine learning or artificial intelligence is enabling breakthroughs in medical imaging. All right. And at the end, I want to leave you with a bunch of possible avenues that you could work on. And these are exciting times. I mean, it is a field which is far from being solved. In fact, it is very much in its infancy. So I think it'll be pretty cool if some of you want to continue working on these aspects and pushing the boundaries even more. Okay. So this is kind of the outline of the talk that I have. I'll talk to you about what I consider is the healthcare problem. There are a lot of problems. I'm going to talk about the problem from a biological perspective, and I will show you, or I will try to discuss with you what do I mean by precision and personalized medicine and where we should go with this. Uh, then I'll talk to you a bit about medical imaging. I'm not entirely sure about the background of all of you. Some of you might have heard of or have some experience with medical imaging, some of you not. So I'm going to, briefly talk about what kind of um, uh, what sort of entails this whole genre of medical imaging. And later on, we'll talk about why do we need AI for medical imaging? Why is it important, right? And then sort of the meat of the talk is going to be on many different deep learning applications to medical imaging, some from sort of the medical side, some from the technological side. And later on, we will talk about what are the problems that we are facing currently, what are the limitations. And finally, we'll end with like the future topics, right? This is like where, I want to see the field go and what we can do about it. So I'm not sure do you have the ability to stop and ask questions, or we just go and we'll do the Q&A at the end, right?
I think it's better to keep it for the end.
All right, let's do that. So let's define the healthcare problem, right? The first thing is that, what do you expect from healthcare, right? In a country that provides healthcare, what you really need from your doctors is they listen to you and they provide a solution to your problem, which is specific to you. Right? Now, how is that actually a reality? How much of that is a reality? And how can we make that a reality going forward? Right? So nowadays you see that everything is being personalized, right? So what you see here is that you're mostly, that all of us agree on that you're mostly your genes, right? But then your genes have a lot of say on what kind of person you're going to be or what kind of uh, problems or health issues you're going to have. But a lot of it is also triggered by the environment that you are in. So there are a lot of studies done on like what they call twin studies, where the twins, for some reason or the other, were living in different households or different environments, different countries, and they showed very different health problems. So you have the same genes, but you have the different environments. And then on the right, you see these different arrows depicting the personal choices you make, the kind of decisions that you make in life. And all these three together result in issues or the way you are. Now, which means that, in an ideal world, let's assume like you're in a perfect world where all this is possible. What I mean by all this is that if you go with a problem, they know all your history, they know your genetic information, they know where you live or in what kind of environment you live, and they know all the choices you made. Like, did you smoke when you were young? Did you drink a lot? Did you do drugs? And so on and so forth. They know all this about the choices you made in life. And combining all this, they tailor a solution which is just perfect for you, right? And that obviously would be the most effective. But we are far from that, right? We are far from having that kind of capabilities. So what we have now is like a dichotomy between personalized medicine and what we call precision medicine. Now, these are terms often used interchangeably in medicine, but they're actually quite different and they have slightly different meanings. So on the left is what I was talking about, that you are an individual and you take all kinds of recordings about you. You take your genetic information, you take your heart rate, your cholesterol, all the things that you can, measure about yourself, your blood test. You can imagine whatever, all the imaging and the medical imaging that we're going to talk about, you do it on that person. And uh, let's say a doctor or a dashboard recommends what to do for a particular ailment. But more, what is actually a reality is what you see on the right. What you see on the right is essentially a depiction of the fact that uh, it might be impossible to give you very tailored, personalized like information on what to do about your problems. But over time, we could cluster people who is like you, right? Like, for example, your same age group, your same sex, your same ethnicity. And let's say that uh, if you can tailor solutions, healthcare solutions to that particular group of people, then you're already much better off than what we have now. What we have now is like, you have a headache or you have a fever, they say, yeah, you just go and buy a medicine, just buy Crocin or uh, ibuprofen or whatever, right? And then you're done. So that is like a solution for the entire population. Doesn't matter who you are, you know, approximately what age you are. You're an adult from 18 to 80. They recommend the same thing. Now, precision medicine is about grouping people together, looking at connections between people. Are they similar people? Are they similar in many different, similarity between people? This is also a different, like I mentioned, right? It's the same sex, same age group, same ethnicity, same food habits. You can use many of these attributes for a person, and then you can cluster them. So this itself can be a machine learning problem, right? So you get a bunch of people, and then you cluster them based on their feature vector. The feature vector could comprise of all these different attributes that I just mentioned. And now what you do is now you design a medicine, an intervention, or a diagnosis for that group. So everybody in that group can have the same kind of medicine, but in a different group, you can tailor the medicine differently. And this is more scalable, right? Because this is possible, given that the number of groups are not too many, right? And the other end of the spectrum is personalized medicine, when the group becomes smaller and smaller, and you're the only one left in the group, right? And this is what we want to achieve. We want to achieve personalization, but we are starting to go from what we have now, which is like broad spectrum, uh, you know, healthcare, to precision medicine, then finally to personalized medicine. So this is the journey we want to take eventually. This is the big vision that people have. Uh, so how does medical imaging play a role? So precision medicine, like I mentioned before, has a lot of attributes, right? Genetics plays the biggest role, and that's a whole different talk on how AI and genetics can come together. There's a huge amount of work being done there, and that's super exciting because it's a fascinating field, actually with a lot of data. So that's also interesting. But there are different attributes. So there's simple clinical diagnostics. There is like blood tests and cholesterol and all that. There's genetics, and imaging forms one of the biggest pillars in going towards precision or personalized medicine. Okay. Now, when we talk about medical imaging, I want to give you a few examples of why it's interesting to do imaging and what are the different kinds of images that you have. So, one of the obvious and the most used reasons for medical imaging is detecting abnormalities. So in this sort of collage, what you're seeing is different kinds of medical images. So you start on the left from some sort of skin disease to brain images with tomography of the lungs, to CT scans, to X-rays, uh, to histopathology, the cancer cells, to eyes, so to uh, do retinopathy and all this sorts of. There's a whole plethora. Every part of your body essentially can be imaged, and they can be imaged by multiple modalities. So reasons why you image, as I said, detecting abnormalities. So you need experts to look at the image and say, oh, this doesn't look really like it's supposed to look, and therefore there's a problem. A classic example is brain tumors. When you take an image, you clearly see a tumor popping up, and they say, oh, there's a problem. The second reason why you want to do uh, imaging would be planning a surgery. So a surgeon needs to know where exactly is the tumor. So the better you can do the imaging, the better he can plan. So it's not like you just cut open the brain, right? So you need to know where to cut to have the minimal damage to other parts of the brain or other parts of the body. And to plan out that whole surgery, they do a bunch of imaging. Okay, things are getting more sophisticated now. People are using these Microsoft Lens and augmented reality, which if you wear these gadgets on your head and you're on the operating theater, you can actually see in real time where the tumor is through the body. So as you're cutting, your knife is superimposed on what you actually see. So, and that's pretty cool. So then you can, you get real-time feedback of how you're doing a particular surgery. Then prognosis. Prognosis is also extremely important. For example, I can give you from brain imaging is that when we look at the brain, sometimes uh, you can start to see shrinkages in certain parts of the brain or certain uh, areas of the brain. And these shrinkages sometimes are just due to aging. As you age, the brain shrinks. But if the shrinkage is totally abnormal, that's an indication, for example, of Alzheimer's. So using those kind of images, we can make a prognosis. Is this person going to be a full-fledged Alzheimer's patient in the next 5 years? Okay. And there the objective is the earlier you can do the prognosis, the better it is, because then we can plan out what to do with this person, how to treat this person. And the earlier we treat a person, the better is the outcome, right? Treatment planning is also very important. Is that, for example, in psychiatric images, so if you have depression is becoming a massive problem all around the world. And it's a lot of money spent on depression uh, and depression related problems. So in depression, there are many different ways to treat a depressed patient, going from what they call CBT, or a sort of cognitive behavioral therapy, where people try to have different sessions with the person, just talk to the person, all the way to the other extreme of deep brain stimulation. That is, they put an electrode into the person and then stimulate certain parts of the brain that will sort of change their mood immediately. And it works, but we need to know whether to bring that person all the way to the surgery, or there are other ways or pharmacological ways of trying to treat that person. Now, nobody knows what to do. So what we do now is random. So randomized control trials, RCTs. They take a bunch of people, put them in a certain kind of group. Other people put them in different kind of group. They do different intervention and see which works. If it doesn't work, they switch the group and stuff like that. So that's not really good for the patient. You want to know beforehand what is going to work. And one of the ways to do that is to build what is known as a biomarker, that when you do the imaging of the brain, for example, I can say, oh, this kind of brain that requires this kind of treatment, right? And finally, imaging is the tool that we use pretty much for research to understand how to generate these biomarkers, or sometimes for pure research, just to understand how the brain works, right? And we'll get into some of these aspects as we go on. The other issue is that we have plenty of different ways of getting images. So when you do deep learning or machine learning, so typically you deal with RGB images. They come from different kinds of cameras, but at the end, they're RGB images, right? And then you can build uh, algorithms based on popular data sets and use those algorithms, for example, like the pre-trained ImageNet or the COCO dataset, and so on and so forth. But medical imaging is quite different because they're not RGB images typically, right? They can be whatever, depending on the modality. You can start with something like an MRI or fMRI machine. Then you can have sort of an X-ray machine, which is, grayscales at different intensities. You can have a retinopathy, which is kind of uh, RGB, but has different meanings to it. Then you have two-photon imaging, which is which is actually a time series which is generated. Then you have gene sequencing machines, which creates images which are quite different. So you have all these different technologies just to get data. So it's when you build an algorithm for a particular medical imaging output, it really depends on what you're looking at because they do have very different properties. And now you sort of understand already from the story that I was trying to tell you that why we actually need AI, right? It's when do we need AI? When you need AI, when like humans really need help, right? And humans need help in many different aspects when it considered when medical imaging is concerned, right? So, for example, one of the issues is that the problems we are dealing with is extremely high dimensional. So one of those brain images is a million dimensional, just one brain image, right? So a million dimension uh, per data set, per data point, right? So we're working at very high dimensions. So usually what radiologists do, for example, if you get an MRI of the brain, is they cannot look at 3D structure together. It doesn't make sense. So they take 2D slices and slice. They slice the brain into in software, not real person. They slice the brain into like 60 or 70 different slices. They look at each slice, right? Now you can imagine what the problem with that is. When you look at each slice, the abnormalities could exist between one slice now and three slices later, right? And if that is the case, you know, human brains are not meant to keep all the images that you're seeing in your own memory, right? So if there is a problem which is related to slice one and slice seven, by the time you reach slice seven, you forgot what happened in slice one. So radiologists have a really tough time unless it's localized. Sometimes the tumors, they are localized. You can see it in one slice. Then, okay, they can detect it. But this is a very tedious job to go through these things. And as more and more people get their x-rays, this is not scalable. Uh, and this is one of the big problems in radiology. Then what we have is that many times when you do analysis, uh, in order to find a difference between what's normal, what's not, you need to do something called a group analysis, that you get a bunch of people who are normal and a bunch of people who have some problem like depression or uh, obsessive compulsive disorder or some other issues, and then you do a group analysis. Now, group analysis is great to show that there is a difference, but it doesn't benefit a single person because what you need is to show that there is a change per person, right? So this cannot be done if you do things in groups. And then is the whole aspect of biomarker discovery that we talked about. That is, what is we need to research and find out what is that signature that tells you that somebody has disease A, B, or C. How do you find that signature? Like if I take the images of my brain, can I predict what are the issues that I'm having or going to have? The other issue is speed of inference. So a radiologist typically take, depending on the problem or the parts of the body, they take half an hour to 1 hour sometimes to actually analyze an image. And that's just too slow, right? Can we do high speed inference that goes at real time speed, okay? Because that's important to do kind of on the fly planning. If you want, like I image this part is okay. If I get the feedback immediately, then I know how to image the rest of the brain. So in a guided imaging can be done like that. And for my personal interest of AI in medical imaging is also democratization of the care, right? So these experts who look at the images, they cost a lot of money, right? They're very expensive. Radiologists and doctors who look at images or who provide care, very expensive. So if you have an AI tool that can run uh, on a simple computer or even on an edge device or on your mobile for skin cancer, for example, this is massive, right? So this we can take it to rural parts of many developing countries and just provide state of the art care for people there, which is awesome. And finally, the population is growing, but the personnel that we need with this kind of expertise are not available at the same rate. And that's another issue why we would need AI to sort of help doctors and sort of bridge that gap. So uh, a typical workflow that you envision should look something like this, where doctors and AI work together, where a patient comes in, he or she goes into the scanner, you get image of your brain with different kinds of modalities. Then the doctor, along with the AI, sort of makes some kind of analysis on the images, and both of them together make a decision on what treatment should be given. So the idea is not to replace the doctor, but to give doctor like superpowers so that he can make a more informed, reliable decision. Because at the end, the decision has to be made by the doctor, at least for now, right? Uh, and there comes the technological aspect. So we're going to focus this a lot of things behind getting uh, AI or machine learning to work in the field of medical imaging. Today, we're going to talk specifically about just technology, right? And then towards the end, I'll allude to some of the other problems and how that can be like future avenues to study. So deep learning, I don't know how much further are you in the course, um, and how many of you have sort of uh, background in deep learning. I'm going to sort of explain this very superficially. If you have like questions, specific questions, you can ask me at the end. But my purpose here is also not to teach you deep learning, but to show you what technologies can be used in medical imaging and how it's being helpful. But in case you have specific questions, you can go ahead and ask me. So all of you might just know that, you know, uh, deep learning typically, sorry, again, somebody had switched on their microphone and getting it. Yeah. Okay, good. Thank you. Uh, so, so deep learning, the popular examples that you must have heard is about giving an image, whether it's a cat or a dog or whatever, it classifies as one of them, right? Airplane, cat, dog, dogs, I don't know, different kinds of dogs and so on and so forth. Now, the principle in many of these is still the same. Instead of feeding in cats and dogs, you feed it different sort of medical images, be it histopathology, be it eye sort of retinopathy images, be it X-rays, and then you build some sort of a deep learning machine that goes and extracts features out of this, and then classifies it as one being abnormal or not abnormal, or segments the images, right? So this is basically the premise. And now we'll go into some examples and uh, talk about some details of how this technology can be used, right? I will try to cover like as many different like possibilities, but there's going to be, you know, I'm just touching the tip of the iceberg here. So, for example, one of the most popular cases was it was in 2017 actually, when people actually Google led this effort to train an algorithm that can detect skin cancer. So for this, they used the inception network. So when I talk about things like inception, if it doesn't make sense, so these deep learning models, when they get really popular and they do something really good or win a competition or do good in ImageNet or whatever, they get a nickname, right? They're ResNets and Inception and so on and so forth. So one of those popular networks is something called Inception, and it was from Google. And the Inception network managed to do incredibly well in detecting skin cancer. So here I will first show you how well it did. So here on the left, you see an example of images of different kinds of skin problems. Okay, epidermal lesions, and these are more a bit more dangerous kind of skin cancers. and you can see what is normal and what is problematic. Okay, these are normal on the top and problematic on the bottom. So if you are not an expert, uh, many of them could be interchangeable and you wouldn't notice too much, right? So what they did was something interesting. They they got uh, I forget how many, maybe 6 or 7 like world experts on skin lesions and skin cancer to annotate a bunch of images, right? A bunch of the same images. So one image was annotated by like uh, 7 experts. So you can imagine this creating this data set is quite expensive because they paid all of them. Uh, so and see here you see there's 22 dermatologists. So 7 was the experts that they used, but they used 22 dermatologists on the same images, and each of them annotated images, and you can see them as little dots here. And this is the sort of the ROC curve. Uh, I guess you all know this, the sensitivity and specificity curve. And you want to be in the top right corner in this curve, right? As much as possible. And the blue line is the algorithm for different thresholds. You can create this curve. And the red points are each of the doctors. Now, the average dermatologist is here. Now you'll see that in most cases, the algorithm is really outperforming any single expert. Okay. Now, that's an important thing to keep in mind. It's outperforming any single expert. So here you already see now when people argue, how can AI do better than an expert? Here is the answer for that. The answer is it's like the AI can learn from multiple experts. Okay. And it's almost universally true that a handful of experts is better than a single expert, right? Uh, even in machine learning, there's a term mixture of experts, sort of has the same sort of analogy in real humans as well. If I get a bunch of top doctors to make a decision as a group after discussion, they make a better decision than as an individual, right? It's just averaging of the noise, I suppose. Uh, and the AI really trains on consistent labeling. That means if all the 7 doctors say that there's a problem, the AI thinks there's a problem. Otherwise, it gives a low weight to that kind of label. The labels are noisy, it gives a low weight. So what it ends up doing is mimics the mixture of experts instead of just mimicking a single expert, right? And that's why it's essentially able to do better, right? It's not real magic, but it's the fact that you can use the expertise of multiple people to make the decision. And that's great. And this made a lot of news splash. Uh, in fact, this can be made into an Android app, and you just can place, if you're worried about something, you just place the app on top of your skin, take a picture, and it'll tell you with some pretty good reliability if you have to be worried and go to a doctor, or you can just ignore it. Yeah. Um, and the other aspect, and this is another aspect of medical imaging that's important, especially when we have to convince the doctors or the non-AI expert that something is kind of meaningful. It's not enough to say, oh, we get 95% accuracy, you're done, this is great, just adopt our AI system. It doesn't work like that. What you got to do is to come up with some kind of visualization, right? And this is a typical, for this kind of 2D images, this is possible, sometimes this is not, is essentially to visualize what these images look like in a much lower dimension. Okay, and this visualization is a popular kind called t-SNE, at a stochastic neighborhood embedding, it's called. And this sort of maintains relative distances between, sorry, uh, relative distances between these high dimensional images in a low dimension, in this case 2D. And when you color it, you see that it's managed to cluster all sort of certain kinds of carcinomas in one part, the ones that are normal in the other, and different kinds of cancers are sort of clustered differently, which means that it has sort of learned something which we call is semantically meaningful uh, in this space. Okay, and this will start to convince others that okay, it is learning something which is interesting. Okay, so this was an example, a very popular example of how AI helped in making skin cancer more accessible. Like I said, it can be on an Android app, so you don't need to go an expensive hospital to do the diagnosis at expert level. Now, switching away from the health aspects, like I mentioned, and try to touch upon both the actual health or the medical aspect of medical imaging to the technological aspect. Uh, so I guess most of you students of like electrical or computer science, engineering, or so on and so forth, and you're well versed with Fourier transform, right? So a so the way we acquire MRI images is not like taking a picture like what you see on the left. It's actually acquired like we see on the right. So medical images are acquired in Fourier space. Okay, and each acquisition of a scanner, the medical scanner that I showed you, each acquisition is one Fourier component. Now, as you know, the only way to get a good image on the left is to fill up as many Fourier components as possible, and then take inverse Fourier transform from Fourier space to get the real space images that you see on the left, right? Um, but each acquisition of a Fourier point on the right takes time, and there is no way you can fill like millions to infinity of points on the right to get sharper and sharper images. But that you don't want the person to stay in the scanner forever. So a scanner, typically you have 5 to 6 minutes to take a good, decent enough scan. So the way they do this, they used to do this, is something called compressed sensing. It's something that you might have studied in your courses, which is essentially a way in which if you know what the structure looks like, you have an idea of what the important Fourier components are. And instead of sampling all of the Fourier components, you just sample those things that you think are important to reconstruct structures of this scale, right? And in fact, if you do compressed sensing properly, you can also do it. You can also beat microsampling. If typically when you do Fourier transforms, you need to at least sample at twice next frequency, right? So here with compressed sensing, you can beat that. That was the state of the art until, of course, you had some deep learning approaches. So if you look at this problem, this is essentially a deconvolution to, or yeah, the inverse Fourier transform is formed by a convolution of a beam. But essentially, the idea is that this is an inverse problem. I'm giving you this. I need to construct from where it came, right? Now, the inverse problem, some of you might have studied that as well. Typically, you start with a model. You start with something like y equal ax plus n, where y is the output. In this case, you say y is that Fourier component that you see on this side. Say these are the y's. Now y is given by some circular matrix here, let's say representing Fourier transform with some noise, right? So this is your forward model of how you obtain your data. So y is your data? Because that's what is measured, okay? One of the ways to solve what x is what's a map approach, the maximum a posteriori approach, right? Which is if you write it down as a simple Bayes formula, if I have a prior of all possible ways, this is where compressed sensing wins because they have a strong prior on the space of what x is. X is your brain, basically. Okay, y is the Fourier transform of your brain. So you take what the probabilities of different kinds of brains that can be, and what's the probability of obtaining the one you've obtained just now, given that particular brain, okay? And then you just need to maximize this probability. which is essentially, if you do it in a dumb way, is you take many different kinds of brains and each time do the Fourier transform and see if this matches with the Fourier transform you've done. And then the brain that matches the best with this is the brain you want, right? This is like a way of forward modeling your way out to the inverse problem. And you can transform this into an iterative scheme where you say, okay, my xt plus 1, that is, I have an estimate. I can start from x0, which is maybe a circular blob, I don't know. And then you say that I can calculate the probability. So here I need some way of calculating what the probability of the output is given the input. And if I find the gradient of that at a particular time with respect to, actually delta t is a gradient of this expression, that is log p y of x plus log p of x with respect to x at particular time t, that will give you an estimate of how xt plus 1 should look. And you can iterate until you reach convergence, right? Now, this is the explicit formula. Now, if you make a change, thinking that xt plus 1 is your old xt plus some function that depends on your log likelihood, then this function can be parameterized as a neural network. Okay, so what you see is an unrolling of this particular statement, where you start with some x0, and then you have the gradient of this probabilities that's flowing, because given this, you can calculate, like the forward model that I said. But you know what y you have, because this is the measurement you made. And there is a difference. Your probability is not perfect, right? And then you can take the gradient with respect to x to update this, what you call a state function, which will then transform x into x1. And you keep doing this until you reach convergence. And like that, you can solve the inference. So this is called a recurrence inference machine. And it did pretty well in some of the big inverse problem challenges, especially with knee x-rays. So here's an example of how well it performs. So on the left, what you have, this is an example of a corrupted image. So take an image and there's some noise in the image, right? And then you run this through this machine, x0, x1, x2, x3. And slowly you can see that compared to many other methods, that quickly learns what the actual underlying x is, right? So it sort of finds the underlying x, which through a forward process generated this noise, okay? Of course, it helps to know what the forward model is. In case of MRI, you know what the forward model is, so it's easy, right? For the forward model, I mean, is you want to know, if I'm giving you a perfect image, how does my instrument and my observation corrupt this? So this was very much used, the exact same technique was used in radio astronomy as well. In radio astronomy as well, you calculate, you know exactly what the forward model is, because it's also a Fourier transform. And then you can do the inverse to get very sharp images of the sky. So on the right here, you see the same technique applied to MRI. The way you apply that to MRI is like, sorry, what happened here? Apologies. Am I back? Yeah, the way you apply this to MRI is best illustrated. Like I told you before, MRI, you basically have an image. The Fourier transform is this, what you see in the bottom, and you make some kind of sampling P and take the inverse Fourier transform, and that's your actual measurement, right? This is what the measurement process looks like. And the recurrence inference architecture, especially for this particular case, looks like this. Y is the observation you've made in the scanner, which are the Fourier transforms. And on the left, you start with the gradients, which you assume here you've shown for a random xt and xt plus 1. But when you're x0, you start with some kind of total blob or some noise. And slowly, this iteration process, as you reach xt to xt plus 1, slowly keeps improving the efficiency. Oh, so not the efficiency, sorry, the contrast or the quality of the image. And here it might be difficult to see the RIM, the recurrence inference machine, can produce similar uh, quality or even better quality than compressed sensing at about 6 times the speed. See, the advantage now, if you think about it, you put a patient in the scan. Patients usually don't like to go into scanners. Many people are afraid of the scanners. They don't want to go into a machine. But instead of putting them for 6 minutes, you can just put them for 6 minutes or even 30 seconds, and you get the same image. Okay, and this is a big gain mostly for the patient, but also in terms of the economics, right? Running these machines is expensive. So the shorter you can run this machine, the better for the healthcare system in general. So this is one application where you see sort of a RNN, LSTM kind of architecture was helpful in improving the technology used for medical imaging, right? Not the analysis of medical imaging, but the technology of medical imaging, right? And there are other use cases of this sort, but I want to go back to analyzing images again. Now, one of the big problems of medical imaging is also the fact that unlike, you know, normal images of cats and dogs, you can get plenty. You can go out with your own camera and take a bunch of pictures, and you can crowdsource that. And before you know it, you can get 1 million, 10 million, 100 million images of cats and dogs and planes and normal things with your camera, right? But medical images don't come like that. Medical images are very expensive. They are very sparse. So and due to data privacy reasons and so on and so forth, it's difficult to collect even hundreds sometimes of images. Thousands of medical images is already in the big data regime as far as medical imaging is concerned. So we need to think of ways in which we can improve the efficiency of how we analyze medical images. So if I give you only 100 images, can I use deep learning? Typically, you have in mind that deep learning can only be used if you have a million images or at least 10,000 images, 1,000 images, right? If I give you 100, can you use deep learning? So there, what you do is so I'll go through a couple of ways in which you can address this problem. One of the ways you address the problem is by looking and exploiting certain aspects of medical images. For example, uh, they do use this in normal images, but in general, it doesn't make sense. So usually, when we look at pictures of the real world, they, even if you have not thought about it, they have a particular direction, right? Actually, even humans are not good at recognizing other humans, even if it's your relative, if I show you a picture of them upside down, right? So even our human system is built in a way that exploits the fact that we are gravity aligned, okay? We don't float around in space. And many of the CNNs are designed to exploit just translational invariances, okay? Probably when you go through more of these course in this lecture series, you will learn more about CNNs and what is translational invariance and how CNNs exploit it. What I'm trying to tell you is that translation invariance itself is a constraint. Because if you look at medical images, typically, if you look at these three examples that I've shown you, one is skin, one is histopathology, and one is the eye. And they are identical images. They have either been rotated or done up-down flip or left-right flip. Identical images, okay? That means there is some kind of symmetry in these images. There is rotational symmetry. Even if I rotate, the object remains the same, okay? And how can we exploit this? So this is a bit mathematical, but I will show you another illustrative example of what this means. So if you know how CNNs work, or if you learn how CNNs work, it's basically you have a filter that looks for, let's say, edges, like the one here, you see the small one that looks for some kind of edges, and that passes through the image and detects, in this case, vertical edges, okay? Now, a group CNN or group equivariant CNNs, different names for this nowadays. What you do is you not only convolve this kernel with the image, but you do that once, then you rotate the kernel in this particular case by 90 degrees and do the convolution again. Rotate it by 90 degrees and do convolution again, and once more. So with the same kernel, by rotating the kernel 3 times, you will get 3 different feature maps. Sorry, 4 different feature maps, because this is P4. It's like all 4 rotations in 90 degrees, which essentially means that this is a depiction of the feature map on the left, and the bottom, let's say, is the first rotation 0. Then you have 4 different feature maps. So each filter can give you 4 different feature maps. So the advantage of this is something called equivariance. So I will illustrate what is equivariance and what is invariance. So invariance means, like this left, there is an image which is rotating, right? It's rotating continuously. And in the bottom, on the right side, on the bottom, you see that whatever is the rotation, the feature maps at a high level in the neuron, that is at higher layers, basically is not changing. Of course, there is a little bit of changes in here, but the orientation of the face is not changed. So this is called invariance. That means the output feature map is invariant to however the rotation is happening. Whereas the top right, here you see that as I rotate the image, the features are rotating at the same. So this is an equivariant network that is able to do that. Okay, and why is that important? Is that now, going back here, an equivariant network requires far fewer number of parameters, because I can use the same kernel, just rotate the kernel and generate these new feature maps. So if I have a object like this or like this, I just need the same kernel to give me the same, in this case, 4 feature maps. These 4 feature maps, of course, the order of them will be slightly different, will be permuted, but basically for this image or this image, I get the same feature map. Whereas if I had used a normal kernel, normal CNN, and just done it once, the feature map for this will look different from the feature map for this. And then there I have to construct more sort of higher layers to sort of combine that information. By using the symmetry argument, I just rotate this filter and create enough feature map that once I have all these 4 feature maps, these 4 feature maps do not change, whatever the orientation of my image is. And therefore, the number of parameters that you use is like way reduced, right? And why is that important? Because as you might know, Weierstrass theorem or whatever you might have studied, the number of parameters that you need is the number of parameters you need to constrain is sort of proportional to the amount of data that you need. So if you have 1,000,000 parameters, you probably need, without regularization, almost 1,000,000 examples. But here, because the number of parameters are smaller, you need far fewer examples. So one of the examples of this, or the first examples of this in 3D imaging, was detecting what lung nodule detections. So pulmonary nodule detections, these are images of the lungs. Basically, it doesn't matter if this nodule is rotated or not. It's a nodule. It's a nodule. And here you see that in the very small data regime, just give me 30 sort of examples, and I can train this by exploiting the symmetry and immediately get 60%. Okay, it's whatever the metric is. And as you increase, even with tenfold, you're starting to approach extremely, pretty good accuracies. So this is extremely important for medical images because sort of gives you a way of dealing with the shortage of data. Okay, and continuing with this is again for different kind of pathologies, not only like X-rays, but if you look at histopathology, the eyes or whatever, this basically shows how with just a few number of samples, here 8 samples, you achieve like really good performance as compared to. So the other way to do that, of course, is to do data augmentation by rotating the image itself and feeding it to the network again and again. But still, it has to learn that invariance, right? You still need a lot of parameters to do that. So this outperforms networks which have been trained with many more parameters and has been trained using data augmentation with just a few samples. So this is a very useful, interesting, and a technique which will stick around whenever you do medical images. Uh, then I want to switch gears and talk about some of the work that we have been doing, which is on a far more complex problem. That is how to deal with like 4-dimensional data. So I'll explain what functional MRI or fMRI is. So MRI, is that a person goes into the scanner and you take a scan and you get one picture of your brain, right? A 4D. Sorry, is there somebody with the mic open? Hello? Yeah, yeah. The 4D scan is basically a person goes in and every 1 or 2 seconds, depending on the scanner and the protocol, you take an image of the brain. So you continuously take images of the brain, right? And this can result in images like you see on the right, which is either they call it DTI image, which tracks the flow of information across the brain in terms of like white matter densities, or you can get these blue and red things that you see are activations of your brain. So for every time point, you'll get a different activation. So essentially what you get is a 4D signal. So at each time t minus 1, t minus 2, and 3, you get an image of the brain, the whole brain. Now, what this really looks like is if you take a slice and if you take a voxel is a volumetric pixel. Like image is a pixel, a 3D object has a voxel, right? And each voxel has some activation over time, right? So you can imagine there is typically in this box, you have about 1 million voxels, okay? Because we take the brain's resolution about 1 millimeter resolution. And each of them can be mapped or scanned for like 200 time points, 200 seconds, right? About 6 minutes of acquisition time. That's massive amount of data. So we're talking about dimensions which are 200 million. So if you take a typical image, if you take ImageNet or whatever, you have 200 by 200 image, right? So it's like 40,000. This one image is like 200 million images. That's a huge dimension. So sometimes in medical imaging is you have to do what like deep learning aficionados don't like to do, right? Because the whole premise of deep learning is you don't need to do feature engineering. Okay, deep learning will do it for you. Yeah, this is true if you have enough data. But sometimes, like in this case, 200 million dimension, like roughly, even if you want the square root of 200 million kind of data set, that's a huge data set. That's multi-billion dollar acquisition of data. Nobody's going to do that. So we have to find a way to reduce the dimension, right? One way is by using domain knowledge. So the experts in the field can say, what are the ways of reducing the dimension? One way to reduce the dimension is like, okay, I have a 4D image, right? 3D in time, but I will summarize each voxel in time into some value. So I can, how much energy is there in the signal, right? You can do a Fourier transform, look at certain frequency bands and say, and that will be a summary value for that voxel. So you reduce the 4D to a 3D by collapsing along one dimension. That can reduce by a factor of 2 orders of magnitude, which is already a great help, right? That's one way to tackle, again, the large dimensionality issue in medical imaging. The other way is the classic transfer learning. For example, what we have done, so this work also we did with Deepak as well, is that we use something like the UK Biobank. UK Biobank is this massive data set. Massive. It's like 50,000 images. For us, 50,000 is a huge number of medical imaging data that they've taken of people. But some people in the UK, right? But we can use those images and learn some task on it, some simple task like predicting the age, predicting the sex, or something like that. And then we can use what we learned there. That means we can use some of the backbone of the model as a pre-trained model to do the classification of our interest, which is in sort of either Alzheimer's or autism or other kinds of psychiatric problems. Uh, so that way we don't have to train the entire network. We just need to train a few layers, right? And if you're not seeing this, this is also probably what you'll see sometime during your course, something called transfer learning, where you don't fully train the network, but you only train different aspects of this. And we managed to achieve pretty decent results not by training from scratch, but by using a pre-trained task, moving it, and fine-tuning it. Here, we've also used something called graph neural networks, which is also becoming super important and popular in sort of medical imaging, and we've done a bunch of work there. But today, there is no time to go into the details of transfer learning. The other sort of exotic ways to do things nowadays is starting to be that, okay, I don't have enough images, but can I synthesize like images with the right properties so that I can start with a small number and I can generate so many more images to train my other classification algorithms. So one way to do that is using GANs, where, for example, with the different imaging modalities, it doesn't matter what these are, but these are the original images, and we can synthesize things that look similar to that. Okay, and why is this important? It just generates more data, more unique data. And sometimes this is a very interesting use case. So the other big case of medical imaging is to detect tumors, as we discussed before, and tumor segmentation. That is, just like image segmentation, you take an MRI image, pass it through some kind of a neural network, a segmentation network, and for each pixel, it will give you an output of what if it's a tumor or not a tumor, and it can segment the tumor, right? But these kind of images are extremely rare, tumor images, right? So what GANs can do is, let's say we take given a tumor like that, I take a normal brain, which is the background, and put a tumor into the normal brain. Now, you said that you could have done this just like without deep learning, right? But that's not the case, because whenever you put a tumor, the brain around it changes in a particular way. It's not just like superimposing two things. And the GANs are able to recognize what that is. So when the GANs embed the tumors, they also change the morphology around these tumors in a way that is mimicking what a real tumor would look like. And now, suddenly, out of like 10, 20 images, you can generate hundreds of thousands of GAN-generated tumor images, which then you can feed into a segmentation. So segmentation models usually are quite heavy. They require a lot of, they typically use something called a UNet or patchwise CNN and things like that. They require quite some data to get it right. So you can use techniques like this to do segmentation of those networks. So before we end, I touched upon only a few applications, both technology and the healthcare aspects, but there are certainly a lot of the problems. A lot of the problems we've already seen. One I've been mentioning is the scarcity of training data, right? It's just not available so easily, right? And the other issue is just the data quality. High resolution data is difficult to obtain, and high resolution data means high dimensionality. So we have to think about ways of reducing dimensionality in smart ways so that you keep the information that you need to diagnose or prognose something. And annotation. So annotation, you think is easy, right? But it's not, because one is for certain diseases, it's easy. It's easy in the sense that if you have the expert, if you have an expert skin cancer specialist, tumor specialist, it's easy, but that's expensive to get these guys and annotate. So usually annotation is outsourced and the annotation quality is not good. But the more problematic thing is sometimes happens in psychiatry. Psychiatry is it's a strange field, right? So if you're talking about OCD, depression, things like that, many of the experts themselves do not agree between each other exactly what that person is, because some of the problems come together. People who have problems with ADHD, like concentrating, might have some problems of depression because they overlap, because people who have an issue tend to be depressed and so on and so forth. So it's very difficult to disentangle. So would you call that person ADHD or would you call that person depressed? And the experts don't agree. There is the DSM-5 protocols, which you answer a bunch of questions and they try to put you in one class or the other. But the problem is, this is where you go back to the beginning and say, this is not personalized or precision anymore, because you're trying to put people in big buckets like depression and ADHD and so on. But people might be somewhere in between. They might be slightly depressed, but mostly ADHD, or mostly ADHD, slightly depressed, and so on and so forth. And since we don't have that kind of fine-grained labels, there's a lot of label noise. How do you deal with that kind of noise? That's a big problem, because for machine learning, typically you need good labels, especially when the number of data points are low. The other issue is, if you look at something like histopathology, that's the skin cancer, or I mean, not the skin cancer, but other cancers where you look under a stain under a microscope, like one of the images that you've seen before, they look very different when it comes from different centers, because they use different microscopes, different staining, different lighting, and so on and so forth. They look very different. So you can't simply aggregate the data, because uh, you know, the algorithm will start to pick up on things which are not important. They start picking up on the lighting or the color of the stain and so on to make diagnosis, which is wrong, right? That's a big problem to aggregate data. The other issue is data privacy. Data privacy is becoming bigger and bigger problem. Uh, Also, I mean, here in Europe, it's been a problem for a while, and it's become much more strict in the last year with GDPR. And the situation is the same throughout the world these days. So if a center doesn't want to give you data, what do you do? Then comes, we can do all the research we want, right? Get the perfect model and so on and so forth. But if you can't put it to use, there's no point in working in this field, right? This is right. If you cannot make sure that it's helpful to somebody, um, that brings us to deployment, right? So how do you actually put this? How do you actually integrate this into the system, the hospital? Uh, then finally, it's a less thought about but equally important aspect is that many doctors, for good reason, are skeptical about AI, right? They are not sure. They're like, what is this thing that is trying to do what we are trying to do? They're not very happy about it, right? And also, they're not confident that it's going to do a right job. So would you trust an AI to make diagnosis? And that requires a change in perception in our society to change it sort of intrinsically. And but on the other hand, all these bottlenecks and limitations lead to future opportunities. This is where you guys can make a difference, right? Starting from fundamental algorithmic contributions to real life contributions. Like if you think of using some of these to make your own startup, what would you be thinking about? One issue is, like I said, they in many systems in manufacturing, so on and so forth, they call it human in the loop system. In our case, we can call it doctor or radiologist in the loop systems. How do you integrate all these fancy stuff that you've done into the workflow of a hospital? Hospital have their own closed software systems that they've been archaic systems that they've been using on Windows for like 30 years. How do you put the algorithm and sort of, they're not going to stop using what they're using and look at your dashboard, right? They want to continue using the system that they've been working on. How do you put that together? How do you work on small data settings? So we've looked at exploiting symmetries. Are there any other ways to do it? We have looked at feature engineering, GANs, and so on, right? are there other smart ways of doing very well in small data setting? Noisy labels we spoke about. And one field that I'm working on right now is unsupervised, semi-supervised learning. So we have these large 50,000 image data set. Instead of pre-training it on a particular task, can we learn what is called pretext task with no supervision at all, or very less supervision? So I guess later in the course, you'll also learn about unsupervised techniques. Like clustering, for example, is unsupervised, right? But how do you do unsupervised in deep learning, right? And then privacy concerns, right? Like I mentioned, what if the hospital doesn't want to give you the data? What do you do then? If the hospital doesn't want you to give the data, you can take the algorithm to the hospital, right? That's the principle behind what's called federated learning. That you take the algorithm, you go to a hospital, I'm simplifying it, but you train your algorithm on that data. You get back the weights of your model. You take that to the next hospital, train it there, and so on and so forth, right? It's the basic idea behind taking the algorithm to the hospital, but not taking the data out of the hospital, right? And this is another avenue to work on, which is a good federated algorithm to work on, how to implement it. There are really no companies working on this properly, because mostly not because of the technological restrictions, because Google is doing this at scale. Each time you use your Android or iPhone and do your smart keyboard thingy where you swivel your fingers and it somehow predicts the right word, is using federated learning to update those models, right? But in the hospital setting, it's difficult. You need to make a lot of you need to go through the red tape of a hospital to allow you to sort of implement that algorithm for them. So these are big problems. Inference at low resource. So many of these models are big networks, but like if you want to put that on a MRI machine, or if you want to take that to rural areas, how do you convert those big algorithms to something that is manageable, a model that is quantized, or to basically, or pruned so that it can fit in a small device like a mobile device or an edge device, right? Anomaly detection is a question that's it's a big topic. So how do we so if I give you a bunch of normal images, using that, can you build an algorithm which will detect which is abnormal? Uncertainty estimation is extremely important, which is basically deep learning has a problem that each time it does inference, it's kind of confident about what the output is. And it doesn't give you the simple algorithms don't give you an estimate of how unsure it is about the output is given you. But doctors like to see that, right? Especially when I make a diagnosis, I say, if somebody says you have this particular cancer, you want to be kind of sure, or at least give an estimate of how wrong you could have been. So uncertainty estimation is a big topic. And decision making. So this is also, the last part is extremely important in terms of treatment. So these are called counterfactuals. Like, what if I give the medicine A instead of medicine B? Which is better? So AI can help really with addressing these issues, which we call counterfactual reasoning, causal inference, and so on. And another aspect that I really didn't mention is something called explainable AI. Explainable AI goes into this, our ambition to convince the doctor. So if deep learning or some other algorithm makes a particular decision, you need a set of tools that makes it easy for us humans to understand why it made a particular decision, right? And that's a whole topic in itself that don't have time to cover today. Okay, so I hope I've given you a overview of all, to not all, but a substantial number of different applications of deep learning and machine learning to medical imaging technology, and all the issues we are still facing that require solving. And solving any one of these is a big breakthrough. And you can work on this at a research level or at a level of the industry where you need to find a solution and push it through. Okay? So that's it from my side. And if you want to know more, or if you are interested in working on some of these topics, you can contact Deepak, this email ID, you probably have it, or you can follow me on LinkedIn, Twitter, and keep you updated. Of course, I'll have content from agriculture, astrophysics, and stuff, but I mostly work on medical imaging as well. So that's it. I think now we can do some QA sessions. I hope I didn't take too much time.
Thank you, Rajat, for the exciting presentation. It was quite informative, but at the same time, I think it was quite motivational as well. After this presentation, I guess we will have at least some students who are quite motivated towards using AI for the medical imaging domain, and we see future research from some of the students.
That'll be great,
But
indeed.
yeah, but for now, I would like to invite the students if they have any questions. It would be nice if you can raise your hand in the chat room, and then I can invite you to ask questions one by one. Yes. So just one moment. I'm not seeing. Just one moment. Okay. So I think the first question we have from is Anangsha Bora.
Hello, am I
Hello,
Yeah, we
audible?
can
yes.
hear you. Yeah, you're audible.
When you talked about medical images, that they are not all are not RGB images. You told that the images of the brains are mostly in million dimensions. Can you please explain like how they are in million dimensions? Like I understood how they are in 4 dimension, but like how can I interpret what is this in million dimension?
Okay, sorry. So what I mean by million dimensions there is like, for example, you worked with, I don't know, the classic example is MNIST images, right? So when you do MNIST images, it's a 28 by 28 image, right? The classic image that you start with. And that we typically say is 28 times 28 is 784 dimensions. That's the dimensionality of your input vector if you consider a simple 28 by 28 pixel MNIST image. A medical image is about 100 by 100 by 100. It's a 3D volume. So 100 by 100 by 100, that makes it 1 million voxels. And then you acquire that over like 200 different times. So you have 200 million points. So it is in that sense that it is a 200 dimensional input. Does that make sense?
Yeah, okay. Thank you, sir.
Okay. So any other questions? I saw someone else raising their hand. Somehow it's not showing in the list. Okay. So we have Kostov.
Yes, sir. I want to ask you that you're talking about the inverse problem, and as we know, high dimensional inverse problems are quite lengthy to solve. So I like to know how that RIM model is running and giving you the result in like 6 seconds.
Yeah. So two things. One is that it's not 6 seconds, it's 6 time points, where that six can be, yeah, it can be 6 seconds, but it's just 6 or 7 iterations, right? Now, the reason why it performs so well is if you have a good forward model. That means the forward model tells you how to. So if I start with an assumption of what my signal looks like, how do I get to my observations? So typically what you do is you multiply with your kernel, which is typically a Fourier kernel or some point first function that you use. So if you know how your signal acquisition instrument works, then it's easy because then the problem is highly regularized. Because if I give you an X, I know exactly how to get the Y because it's a deterministic forward function with some noise, of course. But if you don't know what the forward model is, then it will not be solved in like 6 iterations. You need much more data and many more iterations because then you need to learn what the forward model looks like. That's a much more difficult problem. So inverse problems are kind of solvable if you have a good forward model.
Okay. Thank you, sir.
Thanks, Costo. Any further questions? I think we can entertain one or two more questions. So we have Gautami.
Sir, would you please explain the equivariant again for group CNN?
Yeah. So let's see. Either share my screen again or so. The question is, I'm thinking at what level. So imagine a normal CNN. Okay. So are you all aware of the of CNNs and CNN architectures and how they look and what their feature maps look like?
I think the level might vary, but they should be familiar with CNN.
Yeah.
So we can assume
So
that.
basically, the idea is that in CNNs, right? If I have a cat on the top left of the image, and I generate a feature map from that top left image, right? Now, if I move the cat to another part of the image, the feature map also is identical, but shifted to that side of the image. Okay? And that is the property of CNN. It's translationally equivariant. Okay? So if I move my image, my feature maps also move. But if I rotate the image, the feature maps do not rotate in a normal CNN. An equivariant CNN is it varies equally as your input. That's what it really means. So if you move your input, your output also moves, rotates, not just moves, but also rotates in the same way. This is called equivariant networks, right? Normal CNN is equivariant to translation. A group CNN is equivariant also to rotation. Does that make sense of what equivariant means as opposed to invariant?
Yes, sir.
Yeah.
I think equivariance in itself is a quite detailed topic. It might not be possible to give a full explanation here, but there are a lot of videos on YouTube. There are a lot of tutorials. So if you're really interested, I would recommend looking one of those videos. It's a simple concept, but it's a bit more technical to be explained in 1 or 2 minutes. So spare some time and look around a couple of videos. It should be easy to understand.
Yeah, well, I think so. This GIF file that I showed you explains pretty much an equivariant network. You see, what you see on the right is the output of a certain layer of the CNN on top, right? You start with many layer 1 to 3, is the output of a layer on top. And what it means is that if you rotate the image in an equivariant network, the features are also rotating along with your input. But in a normal CNN, that doesn't happen. It does all kinds of convolutions and pooling and max pooling and so on to make sure that whatever is like an image which is upside down, it does all kinds of transformation to make it straight face. Whereas an equivariant network doesn't care. If it's rotated, it'll give you a rotated face. So in some sense, you can also think of that as a bit, how do you say, philosophically, that an equivariant network has sort of understood better what a phase is. Because if my face is rotating, we understand that the face is rotating. We don't try to, rotate the face and do all kinds of complex geometries in our head to make it look like this, right? And you can imagine that taking a face which is upside down like this and transforming it to something that doesn't change at all requires a lot more parameters. You have to think about all the reflections, the rotations that the network has to do to reach the stage. Whereas the equivariant network is like a face is a face. I don't care. If you rotate it, I also rotate. In that sense, the number of parameters that it requires is far less because it's not trying to reorient the face in a particular direction or something like that. Does that make sense? And equivariance stems out of a whole study of symmetry. And symmetry is a very beautiful topic if you study physics in general. And some of the physicists go as far as to claim that physics is a study of symmetries. So by studying symmetry, you can understand a lot of the laws of physics, like conservation of energies and so on and so forth. And here you use it to understand the dynamics of invariances or equivariances in your input image. I know it's a mouthful, but like Deepak said, there's a lot of good material out there if you want to understand some of the mathematics of it's all based on sort of group theoretical foundations, if that's.
Yeah, thanks, Rajat. I hope it's clear. I think we are also exceeding the time. So, if you have any further questions, I would recommend you can email me and I can forward those questions to Rajat. But for now, I think it's already a lot. So, I would like to also, once again, Rajat already looks tired. uh, so, once again, I would like to thank Rajat for taking out the time for this presentation. It was quite nice. And I also hope the students enjoyed it. And with this, I would like to end this session. Thank you, Rajat.
Thank you. Bye-bye.
Bye.